Our project involves using various microalgae species, specifically Chlorella and Chlamydomonas, to produce lipids adequate for biofuel, pharmaceuticals, and many other applications. In order to achieve this, we had to change the species' genetic makeup. This is the explanation of how we did it. Designing our construct. First, we had to design what characteristics we wanted to introduce into our algae. With extensive reading, we found our promoters, genes, and terminators of interest. Then, we designed our cassettes in silico with the use of a program called BenchLink. Once designed, the DNA was either synthesized and sent to us, or the required plasmids which contained the parts of interest were ordered and the DNA was extracted from them. PCR and purification. Next, we need to isolate and extract a part from the plasmid, a circular piece of DNA, and then multiply it. We did this using PCR, gel electrophoresis, and gel extraction. This step was repeated with a variety of plasmids until we had enough genetic parts to be able to build our constructs. Gibson assembly. Next, we had to assemble the parts into a new construct or cassette. This was executed via Gibson assembly, in which our desired parts and a backbone were combined to be made into a novel plasmid. To multiply the plasmid, we used E. coli. We inserted our newly constructed plasmid into chemical competent DH5 alpha E. coli cells by a procedure known as heat shocking. Once heat shocked, the E. coli were plated on a petri dish containing nutrients allowing them to grow, as well as ampicillin antibiotic. Using ampicillin as a selectable marker ensured only the E. coli cells with our plasmid, which contains ampicillin resistant, were able to replicate. Once incubated at 37 degrees C overnight, we select the transformed E. coli and transfer them to a liquid culture containing LB plus AMP100. These cultures are once again incubated at 37 degrees C overnight and allowed to flourish. Plasmid extraction. The following day, our plasmids are ready for extraction from the E. coli cells. Linearization. Once extracted, we are able to linearize our plasmid using restriction enzymes. Linearization is crucial to allow the insertion of our cassette into the genome of the organism through homologous recombination. Electroporation. Once linearized, our new cassette can be inserted into our microalgae. We use different strains, including a cell wall deficient strain of Chlamydomonas, as well as Chlorella vulgaris and Chlorella kessleri. The insertion was facilitated using electroporation. Once electroporated, the algae were incubated in a 6-well plate overnight for recovery. The following day, they were plated on tap plates supplemented with hygromycin as a selective marker. The transformed cells containing a resistance to the hygromycin gene will survive, all others will not. Then we transfer the survivors to a liquid culture in a 6-well plate so they can grow. Now it is time to determine whether or not we have our novel microalgae species. To check the outcome, we use the following: flow cytometer to check for GFP expression, DNA extraction from our constructs, followed by PCR amplification with sequencing primers and gel electrophoresis. All tests show we got our transformation. Hooray! Now our transformed Chlorella and Chlamydomonas are ready to perform their designed functions. Either in industry to produce biolipids for biofuels or pharmaceuticals, or being a new vector in research.